Hello again. Welcome back to Biology 102 online lectures. Remember, you're never alone. You can email me, you can schedule an appointment to meet with me, you can talk to other students, post things online. So I encourage you not to sit and feel like you're alone in this particular class. So also remember that you need to print out your PowerPoint lecture outlines. Those are on Blackboard. So I would print it 6 slides per page. That way you can fill notes out as we go along, because the note outlines aren't complete notes. You have to take notes as you go along and do drawings as we go along. So today we're going to talk about prokaryotes. So spend a second and think about, what do you know about bacteria? You can stop the recording and or lecture video and write down what you think. So this is the head of a needle, and you can see that the outline of the needle right here. And each of these little creatures here are bacteria. So bacteria are relatively small. Of course, we need a microscope in order to see them, but these are very, very important organisms that we're going to talk about today. So prokaryotes do come in one of 3 shapes. They come in a circular shape, and these are called cocci. They come in kind of what I guess some people describe as a rod shape, and these are called bacilli. And then they come as in terms of a spiral shaped, and these are called spiralia. So we have different shapes, and sometimes these things are stuck together in chains, as you can see here and here. Sometimes they're individual, and sometimes they're just in clusters. So I'm going to make a drawing of a prokaryotic cell for you here in just a second, but we're going to include these particular parts. We're going to include a cell wall. We're going to include the sex pili. We're going to include the flagella and plasma membrane, cytoplasm, and also the nucleoid. So these are some of the parts that bacteria have in common, or prokaryotic cells have in common, that we're going to indicate on our drawing. So you take it home and make sure you do the drawing too. I'll do the best I can drawing on a screen here. So let me take my stylus and see what I can draw here. So bacteria cells have 2 outside parts. They have a cell wall and they have a cell membrane. Okay, so 2 layers, cell wall and cell membrane. Inside they have a region called the nucleoid. It's a region that holds the bacterial chromosome. So this is a circular bacterial chromosome. And so you are probably familiar with cytoplasm, so that would be cytoplasm, and we'll put in a few ribosomes. I'm creating them as little dots that are inside, and those are little protein factories that the cell will use to make protein. Also, some of these prokaryotic cells will have a flagella. This is a whip-like structure that comes out of the plasma membrane and will create movement for the cell. And then others of these prokaryotic cells may have these little extensions that come out. These are almost like little tubes where they can transmit these things called plasmids between cells that are connected via those sex pili. So, and sometimes bacteria or prokaryotic cells even have plasmids. These are little circular pieces of DNA that hold very specific genes. So this is generally what a prokaryotic cell looks like, and you should be able to sketch one on a test. You should be able to sketch and label a prokaryotic cell. Remember, there are study guides for you to look at online and to complete. You can do that by yourself, or you can do that with a partner if you choose to do so. Those are for extra credit, but also it gives you a good indication of what's going to be on the test. So you notice on your chart that you printed out that some of these things are blank. So as you're filling out the blanks, I'll go over it. I did a little chart here to compare and contrast bacteria, archaea, and eukarya. Bacteria and archaea are going to be known as the prokaryotes. So you can see that prokaryotes do not have a nucleus, but eukaryotic cells do have a nucleus. This is a major difference between those two groups. You can see that bacteria and archaea, our prokaryotes, have a circular chromosome, but eukaryotes do not have circular chromosomes. They actually have linear or line-like chromosomes. Histones are proteins that are associated with DNA, and bacteria do not have histones. Archaeans do have histones, and eukaryotes do have histone proteins as well. So you can see there are some differences between bacteria and archaeans. Even though they're both prokaryotes, there are differences between those two. I just put a couple of antibiotics here, streptomycin and chloramphenicol. And so if you notice that bacteria are inhibited by these antibiotics, and so are archaeans, but you can take these antibiotics and not kill yourself because you are not susceptible to the effects of these significantly. Okay, so that's how antibiotics work. They kill some organisms, but they don't kill you, hopefully. In regards to membrane-bound organelles, bacteria do not have membrane-bound organelles like mitochondria, chloroplasts, and things like that, endoplasmic reticulum, Golgi apparatus. Neither do the archaeans, but we do definitely have those in eukaryotic cells. That's a major characteristic difference between prokaryotic cells and eukaryotic cells. Let me slide a picture up over here. So cell wall made of peptidoglycan. Peptidoglycan is a molecule that bacteria make their cell walls out of, but archaeans do not, and eukaryotes do not make their cell walls out of peptidoglycan. This is just showing you the bacteria flagellum. I just think it's one of the most incredible structures. It's just really cool. It's, you know, we think we invented wheels, but you know, there is a wheel here, and then you have an axle here, and it rotates around and around, causing the bacteria to move. There's a little web link there if you want to read about the bacteria flagellum. So prokaryotes do contain a single circular double-stranded chromosome. It has about 4.6 million base pairs, or 4,300 genes, generally speaking. Double-stranded means it has two pieces of DNA that are, making up the double helix. Chromosomes are found in the region called the nucleoid, as we drew in just a second ago, and there is no membrane around the DNA. So there is no nucleus, there's only the nucleoid. Bacteria divide by binary fission. This is where the single one chromosome is duplicated, and then the bacterial splits in half, separating the duplicated genetic material into each individual daughter cell. And a bacterial cell colony is a colony of genetically identical clones. So you'll be growing bacteria with one of your labs. You want to make sure you do that lab early, because it takes a little bit of time for that lab to happen. But hopefully you'll be able to grow some bacteria. And when you look at your bacteria plates, you'll see there's little colonies on them. And each of those little round circular colonies is a clone where one bacterial cell landed on that agar plate, that nutrient plate that you'll be making and growing, and it grew into a genetically formed colony. Everyone in that colony is genetically identical, unless there's some kind of a spontaneous mutation that occurred in one of the individual bacterial cells. So how do bacterial cells reproduce and produce genetic variation without sexual reproduction? Because bacteria don't have sperm cells and egg cells. They literally copy their DNA, split in half, so that their daughter cells are clones of the parent. But there is the ability for them to get genetic variation, so that they can evolve and adapt to changing environmental conditions. So genetic recombination, by definition, is the combining of DNA of two individuals into one individual's genome. So even though bacterial cells don't have sperm cells and egg cells, they do have the ability to create genetic variation. So, humans, we have sperm cells and egg cells, so that all of our progeny are genetically different than the parents that made them. So we have sexual reproduction in eukaryotes, and that's the combination of the sperm cell and egg cell to create a zygote that creates genetic recombination. Bacteria, though, even though they don't have sexual reproduction, they do have 3 different ways that they can combine genetic material to create new varieties or new strains. So the first way is called transformation. Transformation is a process that's sometimes mediated by proteins in the membrane of the bacterial cell. And what these proteins do is they bring in DNA that's just in the environment sitting around. And it incorporates this DNA into the bacterial genome. So if you take it home and you spray your countertops with Lysol and kill all the bacteria, yeah, it kills all the bacteria, but the DNA now is sitting on the surface. So any of that DNA can be brought into other bacterial cells that will land after the cleaning product goes away. So that's called transformation. Transduction is where DNA is transferred by the use of phages, those bacteriophages that we talked about in the last lecture. So these can carry pieces of DNA, these phages can carry pieces of DNA from one cell to another, and it kind of looks like this. So up here we have a bacteriophage, which is represented by, I assume this is a purple kind of DNA, and you can see the bacterial chromosome here is represented by green DNA. So when the bacteriophage lands on the cell, it injects its individual genetic material into the bacterial cell, and then you can see that it scrambles up all of the DNA, bacterial and phage DNA. And notice that most of the time phage DNA arrives in little phages that are being created. But over here, look at that, the bacterial chromosome, a little piece of the bacterial chromosome, that little green piece right there, happens to accidentally get into phage virus. So you can see it's traveling over here, and if it travels and infects a new bacterial cell and it'll incorporate itself into the bacterial chromosome, and maybe it doesn't kill the bacterial chromosome, and now you have genetic variation. So you can see that the bacterial DNA is now incorporated into the bacterium, and so you have genetic recombination. Conjugation is the direct transfer of DNA between 2 joined bacterial cells. Usually this occurs through what we call a plasmid. A plasmid is an extra little piece of circular DNA, and you can see here's a sex pilus right here, and these 2 bacteria are undergoing conjugation, and a little plasmid can actually travel through this straw-like sex pili and transfer genes to another bacterium. So plasmids are those small circular DNA molecules. These are separate from the bacterial chromosome though, so but they can transfer and share genetic material. There are 2 types of plasmids that you need to be familiar with. The F plasmid is a plasmid with a small number of genes that codes for the production of a sex pilus. Sometimes bacteria can't make sex pili, but if it conjugates with another bacterium and that bacterium sends an F plasmid over, it too will be able to have the genetic material, the coded instructions to build a sex pilus. The one that we need to worry about though is the R plasmid. R plasmid, the R stands for resistance. And these plasmids convey genes for antibiotic resistance. I'm pretty sure you're familiar with the use of antibiotics curing bacterial infection, and you're probably familiar with the concept that our antibiotics are becoming less effective to bacteria over time. So antibiotics resistance is pretty scary because you get strep throat now. Today you go get an antibiotic and it destroys it. But if we lose the ability to use antibiotics, a lot of people used to die from strep throat. So antibiotic resistance is a pretty important topic. These R plasmids contain 10 or more genes that may make ineffective 10 or more antibiotics, which is pretty scary. Transposons are mobile pieces of DNA in bacterial chromosomes. These allow plasmids to have very complex genes, and I just wanted to show you that a transposon is a movable piece of DNA that can move from one place in the chromosome to another place, or from the chromosome to the plasmid, and so this is how some genes get into plasmids. Now, because of all this gene transfer, this transduction, transformation, and conjugation, we don't even know what bacteria really are. We don't really call bacteria species per se. We typically call them strains. So, and I just had a little quote here from a microbiologist. Indeed, the exchange of genes among different bacterial cells is so pervasive that the entire bacterial world can be thought of as a single huge multicellular organism. What's crazy about bacteria is that one strain of bacteria, if it receives certain kinds of genes, can actually convert to another strain of bacteria. So we don't really use the word species for bacteria. We do use the word strain for bacteria because of all this transfer of DNA. So describe 3 ways that bacteria can cause genetic recombination. Which ways do you, so out of those ways, which ways do you think scientists doing genetic engineering research would find to be the most useful? So you could stop the computer for a second and think about that, or I can go ahead and tell you that scientists use they use several different ways. They do use transformation, where they put DNA and they can heat shock a bacteria to bring in DNA. We also use plasmids. Plasmids are the workhorse of genetic engineering, so we definitely use a lot of plasmids in order to do genetic engineering, to insert genes into bacteria that we want the bacteria to make. Do you think plasmids and transposons, what do you think plasmids and transposons have in common with viruses? Yes, you're right. They move around just like viruses. They can jump from place to place and act just like viruses. So let's talk about antibiotic resistance just for a second. So antibiotic resistance is the ability of bacteria to evolve defenses against antibiotics, thus making the antibiotics useless to them. Now, just keep in mind that bacteria can reproduce about 16 million offspring in 24 hours, theoretically. And in an environment of antibiotics, so if you take antibiotics or expose bacteria to antibiotics, due to natural selection, there's always going to be resistant forms that are going to be exposed to the antibiotics. Now, if you have an environment free of antibiotics, bacteria typically lose resistance to these chemicals because it is a cost to maintaining genes for resistance to antibiotics. And if you don't need the genes, you don't have the genes. So it's an energy, it's just the efficiency using energy. So here's a colony of bacteria right here. And let's say we introduce an antibiotic. So you can see that out of that whole colony of bacteria, there may be only one bacterium that is resistant to the drug. But if you keep exposing all these things to antibiotics, notice that in the future, all of the progeny will be resistant to the antibiotics. So if we don't have an environment of antibiotics, then that particular individual cell that has resistance is going to be very rare because remember, it costs energy to maintain those genes for antibiotic resistance. So we want to try to not use antibiotics so that we create an environment that selects for less harmful bacteria. Now, if you ever go to the hospital and you pick up an antibiotic resistant bacterium, what will happen is you'll ingest it because you didn't wash your hands after using the elevator or whatever. Through conjugation, it will transfer those antibiotic genes to other bacteria, and now you have a harmful bacterium that is reproducing in your body. So if you ever go to the hospital, I'll call you, I'll text you, I'll email you, but I'm not coming to see you because I don't want to inherit or to pick up those antibiotic resistant microorganisms. So the place that has the most dangerous microorganisms would be places like nursing homes and hospitals, because this is where sick people are and where they go and where people don't have immune systems. And so that's where you're going to find those particular things. There is a little antibiotic resistance video that you can watch. You can watch it through this PowerPoint, or you can watch it through, I put it on Blackboard for you to look at. Let me know what you think about that. So what are we doing to exacerbate the problem of antibiotic resistance? Well, physicians, every time a patient comes to the doctor, they want antibiotics. Even though the disease may be a viral disease, People would just ask for antibiotics because they think that's going to cure all diseases. But remember, from last lecture, we found out that antibiotics do not work against the viruses. They only work against living cells. Therefore, if you have a viral infection, no reason to have antibiotics. It's actually dangerous for you to overtake antibiotics because you're creating an environment in your body of antibiotic resistance. So oftentimes patients don't comply with the directions for taking antibiotics, and that could be dangerous too. So if the antibiotics say take it for 7 days, take it for 7 days. If it's 10 days, take it for 10 days. Make sure you follow the procedures and the rules for taking that. In other countries, you don't even need a prescription to get antibiotics. You can just buy them over the counter, and that's dangerous because everybody's taking lots of antibiotics and creating more antibiotic resistance. So immune system diseases do not allow a person to kill bacteria naturally, so these folks have to take lots and lots of antibiotics, and that creates a situation of antibiotic resistance. And hospitals just being dirty and not maintaining a sterile environment also fosters the development of antibiotic resistance as well. Now, one place that you never really kind of thought about antibiotics maybe is agriculture. So when we grow animals like chickens and cows, a lot of times we grow these in places where they're really tightly contained, industrial kinds of settings. And 84%, this is pretty staggering, 84% of all the antibiotics used in the United States are used to promote growth. That is, not to cure disease, but to promote growth in farm animals. Okay, now some of those antibiotics are used to treat infections, but a lot of them are used to grow animal muscle tissue faster. Remember, when you eat meat, you're eating muscle tissue, and if you want to make lots of profit, then you want to promote the growth of your animals, and doing antibiotic treatments actually promotes the growth of that. So hopefully one day we'll wisen up and maybe stop the use of this stuff in agriculture as much as we do. So what role, if any, have you played in the increase in antibiotic resistance of bacteria? Should antibiotics be banned except to treat illness that are only illnesses caused by bacteria? And should antibiotics be used or be banned, excuse me, for the use of them in agriculture? So what do you think about that? Some solutions that have been proposed would be an advocate for yourself and family members when in the hospital and doctor's offices. If it's not bacterial, don't take an antibiotic. So organic farming uses no antibiotics. That's what they, part of the definition of organic means that you don't use chemicals, steroids, or other chemicals. So, maybe if we start buying more organically grown meats and things, that will cause other farmers not to use the antibiotics. Of course, just reducing the use of antibiotics, better sterilization practices. We do irradiate food. That is, we take radiation and expose meat and other products to this radiation, and it kills all the bacteria, hopefully, and viruses on the food, and so our food is cleaner when it's irradiated. Now, it doesn't leave radiation in the food. It only uses the radiation to nuke the bacteria and viruses that are on the surface of the meats. So making phages, genetically engineering phages, which are viruses that kill bacteria, has been done in other places in the world. We don't actively do that here in the United States that much yet, but it's coming. Development of new drugs is always working, and education of people about this problem is going to help reduce the problem, hopefully. When antibiotics go away, we go back to living in the Middle Ages, and that was not a pretty time to live. So we want to make sure we keep those antibiotics useful for us as long as we can. What I want to do now is talk a little bit about the biodiversity. That's a word that means the different kinds of strains that are around the world. And I want to talk about prokaryotes. I'm going to start talking about bacteria first, and then I'll talk about the archaeans. I always want you to remember, it's a big concept of mine that I think is really important, that you understand that it's the small things that rule the world. This is really true. We think humans rule the world. We think we're different than nature, but we are interdependent upon all the small things helping us to, produce oxygen and forming food chains and forming chemicals that we use and being things that other things eat. So recycling and decomposing dead stuff. The small things truly do rule the world. So I want you to appreciate that during the course of this class. So back a long time ago, there was a universal ancestor of all living things. We think this existed because if we look at all the modern day creatures, the eukaryotes, the archaeans, and the bacteria, they all have things that they share in common. For example, they all do glycolysis, they all have DNA, they all have ribosomes, they all have cell membranes. They even have genes that are in common with each other. So, but they are different as well. So over time, you can see in this little phylogenetic tree that over time they diversified into eukaryotes and archaeans and bacteria. So things have evolved over time. So let's talk about first of all this grouping called domain. There are three main domains. You can see there are eukaryotes, the archaeans, and the bacteria. These are major groupings that we use in classification. So we'll talk about bacteria first, and the first grouping I want to talk to you about is the proteobacteria. These are also known as gram-negative bacteria. There is a gram stain test that we do at the hospitals to see what kind of bacteria people might be infected with. And some bacteria are gram-positive, and some bacteria are gram-negative. If you ever get a chance to take a microbiology class, I think you'd be very interested in that. It's fascinating, growing bacteria and looking at them under the microscope. So these proteobacteria, one of them that's really super important is rhizobium, and these live in the roots of plants. If you look at these little nodules over here, these little pinkish nodules on these roots, these are infections where the plant has invited the bacteria to come in and infect it. And what the rhizobium does is it lives in those roots of those plants, and it provides nitrogen for the plants, and the plants provide a home and food for the bacterium. So that's a mutualistic relationship between those organisms. Nitrosomonas, these are bacteria that live in soil, and they are basically part of the nitrogen cycle. So they recycle nitrogen and bring nitrogen into the soil so that plants can grow and that we can kill plants and eat plants and survive. Now, there are good guys and bad guys in all of these bacteria we're going to talk about. A couple of your bad guys would be your Salmonella, your E. coli, and your cholera. These are disease-causing proteobacteria. And relatively recently, we discovered this bacterium called Helicobacter pylori. This is what it looks like down here. It's kind of cool looking. Look at those flagella. And so what these particular organisms do, this Helicobacter pylori, is they will digest into the wall of your stomach and they will cause ulcers to form. Most ulcers nowadays are thought to be caused by bacterial infections and not stress, alcohol, aspirin, or dietary kinds of things, or stress. So, so that's something that we're seeing today is that people that have stomach ulcers, they check them to see if they have the bacterium, and then they give them an antibiotic to cure that. Oddly enough, the person that discovered that actually drank the bacteria, caused himself to develop an ulcer, and then took an antibiotic to cure the ulcer. So he went on to win the Nobel Prize in medicine. I don't advocate that you do that if you get into science, though. It sounds kind of crazy. So other kinds of bacteria would include the chlamydias. The most common sexually transmitted infection in the United States is chlamydia. It's common, it's doing very well, and it spreads very easily from humans to humans through unprotected sexual intercourse. I used an old term, sorry about that, sexually transmitted disease. We're trying to use sexually transmitted infections now to take away the stigma, because it is an infection just like any other infection, although it is sexually transmitted. So your spirochetes include things like syphilis and Lyme disease. These are really bad guys, and this is what their shape actually looks like. I worry about these guys all the time because I'm a field researcher. I do a lot of research on reptiles and amphibians, so I'm always having ticks on me all the time. So I've actually had Rocky Mountain spotted fever. And my wife right now has alpha-gal, which means she's allergic to any product that comes from a mammal and milk, and cheese. She was bitten by a tick. The tick injected into her proteins from a deer and sugars from a deer that they were feeding on, and she became allergic to the mammalian meat and milk products. This particular tick here, the deer tick, is responsible for creating this little bullseye rash here. So the tick would have bitten right here in the center, and over time a rash developed, and that's a major symptom of Lyme disease. Although some people don't have it or ever recognize that they actually have that after being bitten by a tick. Now, if you catch Lyme disease early, you can take antibiotics and cure yourself, just like you can most of the bacterial infections transmitted by ticks. But there are some viral infections transmitted by ticks that you can't use antibiotics for, as you already know. Some of them are really nasty, and these tick-borne diseases seem to be becoming more common and widespread. Maybe it's because we're looking for them and being able to detect them a little bit more than in the past. So gram-positive bacteria include things like strep, botulism, anthrax, tuberculosis, and leprosy. And those are bad guys for sure. They killed a lot of people. So strep throat, most of you have had that before. They used to kill people. I mean, they used to kill people before we had antibiotics. So there are some bad guys. There are some really good guys, though. There are some ones that decompose things, and there's others that make antibiotics that we use to treat bacterial diseases. So bacteria do, fight each other, and they use antibiotics in that warfare, and we can harvest those antibiotics and use them for our purposes. A really good guy would be your cyanobacteria. These are photosynthetic bacteria, and they create oxygen for us to breathe. Now, the world is 3/4 water. Earth is huge, and it's mainly water. And these little creatures live in water, and they live by the trillions and trillions and trillions, and they create huge amounts of oxygen. The cyanobacteria, along with the algae, probably make 70% of all our oxygen that we actually breathe. So theoretically, you probably could cut down all the trees on Earth and still have enough oxygen to breathe, as long as there are organisms in the ocean. We want to be real careful, because the ocean, we want to protect it, and if we don't protect the ocean, we don't protect our oxygen supply. So photosynthetic bacteria. Sometimes these do cause problems, though. They can grow in large amounts when humans use lots of fertilizer that flows into the water. The fertilizer is food for these organisms, and they can grow in huge amounts and create algae blooms. These algae blooms can be smelly. They can be toxic oftentimes to wildlife in the ocean. So we have to manage these things and protect them, and definitely not let them overgrow if we can help it. So those were some of the bacteria. Now, what I want to do is talk about now domain archaea. Archaeans are really interesting organisms, and you probably aren't too familiar with them. So let me share a little bit about them. Some of them are extremophiles, and not all of them are, but one group of them are called methanogens. And these methanogens produce methane as a waste product of their respiration. So you produce carbon dioxide when you do respiration. These creatures create methane gas. So they live in marshes and swamps in the intestines of animals, and they have an interesting characteristic that they die in the presence of oxygen. So they live in places where there's no oxygen. Swamps, intestines, all these places are going to have low oxygen or no oxygen, and they grow. And if you've ever been to a swamp before, or gone to a place that has tides and it's kind of swampy, it does smell like methane. So it does have methane gas smell. And some swamps produce so much methane you can light a flame and it will actually create an explosion. So one thing about these creatures that's a little bit significant is that they do exist in the intestines of herbivores like cows, and they do produce a huge amount of methane gas. So methane is a global warming gas, and methane, in addition to carbon dioxide, are two gases thought to be increasing in amount and are leading to the warming of the Earth. There are also creatures that are archaeans that are known as extreme halophils, and these are creatures that live in extremely salty areas. So they can live in places like this. These are called salt evaporation ponds. They pour water in these ponds, and these ponds are typically in the desert, and then they let the ponds dry up, and you can see the white around the edge is salt that's forming. But these little organisms live in very salty areas, and especially salt evaporation ponds. So they're kind of cool. They have different colors in this particular diagram. There are also extreme thermophiles. These live in very hot environments like deep sea vents, and these are where underwater volcanoes are erupting, and hot water springs. So this particular picture here is of a spring in Yellowstone National Park. You can see there's different layers of colors because there's different strains of these archaeans that live in different temperatures. So at the very hottest temperatures, you really don't have too much of anything living, but as the water moves away from the center and cools, there are different temperature gradients that different archaeans actually live in. I don't know if your teacher went over it in Biology 101, but there is a technique called PCR, polymerase chain reaction, where we can take small amounts of DNA and amplify it into lots and lots and lots of DNA. In order to do that, we have to have a process that heats and cools and heats and cools and heats and cools, and we actually use a polymerase from organisms that we got from a hot environment. And those polymerases can survive the heating and cooling process of PCR. So that's probably a billion-dollar industry created from something that we learned from studying bacteria that live in boiling hot pools. Now, these prokaryotes don't just eat food, but they do all different kinds of ways that they do their metabolism in. And let me say that a little bit more clearly. So we do have some prokaryotes that are photoautotrophs. These are organisms that harvest light energy and use carbon dioxide to make organic compounds. So that's very similar to what you're familiar with as photosynthesis. But then there are chemoautotrophs, and these are organisms, are prokaryotes that harvest energy from inorganic substances, and they use carbon dioxide to make organic compounds. So there are lots of different inorganic substances like hydrogen sulfide that these organisms can use, and they still use carbon dioxide, and they can make organic compounds from that. There are also photoheterotrophs. These generate ATP using light, but they have to eat organic forms of carbon. So they actually have to go and eat organic forms of carbon, maybe decaying, dying organisms, but they do use light to make ATP. That's really a cool organism. And then there are chemoheterotrophs, and these are just organisms that consume organic molecules, both for energy and for their carbon source, so they can grow new molecules or make new molecules for their body. So these are very metabolically diverse groups. And what I mean by that, metabolically diverse, all animals are chemoheterotrophs, and we don't do any other thing. All plants, for the most part, except for true parasitic plants, are photoautotrophs. So prokaryotic cells have multiple ways of metabolically undergoing different processes. So there are extremophiles in this archaean group that love acid. There are some that live in very basic environments. There are some that like extreme heat. Hyperthermophiles really like heat. Psychrophiles like cold temperatures. Halophils like salt. And osmophils can live in high sugar concentrations. So how are prokaryotes important? They're important for many ways. They're important in chemical recycling. They aid in decomposition. They aid in nitrogen fixation, so they take nitrogen from the air and make nitrogenous compounds that other organisms can use. They undergo photosynthesis, and they're members of food webs, so other things eat them. They also form symbiotic relationships like Rhizobium did in the roots of legumes, which are the bean family. They live on our skin and in commensalistic forms where they eat skin cells, but they don't really help us in any way. They don't hurt us in any way. Some of them are parasitic. Some of them cause, you know, some serious human diseases. And we're beginning to use these organisms in what we call bioremediation, which is the removal of pollutants from air, water, and soil, and sewage and oil spills to clean up the environment. So they're genetically modified to do that, and oftentimes. And we produce yogurt, cheese, and antibiotics. So commercial production of products is also using bacteria. So pathogenic bacteria cause half of human deaths, so this is definitely why they're important as well. Some of these organisms create exotoxins that they actually dump into the environment or into your bloodstream or body juices. Cholera toxin would be an example of that. Some of them produce endotoxins, which are just components of their membranes or parts of their cells that when they're destroyed, they release these toxins into your body, and they can lead to you know, toxic shock syndrome, fever, inflammation, or swelling. Biofilms is a really important concept that I want you to get familiar with. Biofilms are going to be up and coming. They're really important. They're going to be a billion-dollar industry trying to figure out how to tackle and destroy them. So the microbial community, held together by polysaccharides and proteins secreted by the organism, creates what we call a biofilm. And you've all seen these biofilms before. So you know, we have to basically clean our houses because of biofilms. So they coat our sinks, they coat toilets. So if you ever seen your toilet getting darker and darker color after, you know, a week or two of after cleaning, these are biofilms. Cutting boards are biofilms, and so if you've ever walked on a stream rock before and slipped and fell, you can curse the biofilm, because the biofilm is the organisms that got you. It makes it very slippery rocks. So these biofilms coat all of our pipes inside of our house, and they're all over the surface of everything. Biofilms are everywhere. They're on your teeth. I mean, do you go to the dentist often? Because, you know, that's why we go to the dentist, is to get them to polish our teeth and get rid of those biofilms. Biofilms are important because they can contaminate food. They can grow inside of canned foods. They coat our teeth and cause dental cavities. They clog our pipes, but they also serve many useful purposes in natural ecosystems. And we have biofilms on our body as well, so they definitely are useful for us as well. So this is just a picture showing you how bacteria will adhere to a surface. It'll begin to grow. It'll begin to secrete polysaccharides and proteins on the outside surface. It'll form a little community. There will be more species that will come in, and eventually they will begin to form these little detaching parts and go platonic and form other biofilms in other areas. One place you don't want a biofilm is in your joints. So sometimes bacteria can shield themselves from antibiotics by creating biofilms. So these are really important to understand. I put a video here about bacterial communities, and I find it to be really interesting. I really like the speaker as well. She's an excellent speaker, and I think you'll find what she says to be exciting. Take a look at that. I put it online for you as a link. So microbiome is a current topic we talk about when we talk about prokaryotic cells. This is the community of organisms that share the surfaces of our bodies both inside and out. You have trillions, literally trillions of bacteria living inside and outside of your body. And this is what they look like. Aren't they cool? And this is just basically, this is a splinter of wood right here, and false colored bacteria all over the surface of it. It's amazing, amazing photography. To learn more about the microbiome, I did put this invisible universe of the microbiome. You can look at that in the lecture, or you can look at that online. I posted it online for you to look at. So that completes this particular lecture. Remember, you're never alone. Make sure you email and you talk to other students and go on those discussion boards and discuss things that you are finding interesting. If you're an introvert, email me. Let me know what you're thinking about some of these things. So if you have questions, let me know. Make sure you work on your study guide. Make sure you keep up with your due dates. Make sure you keep current. Don't wait till the last minute to do things. If you have any questions, any problems, let me know, and we'll definitely help you out. I hope you have a good day.